guys, this is Malinki. Welcome back to my channel, Voice of Malinki. Today we will talk about digestion of food. And if you are new in my channel, please subscribe my channel. And if you like my video, please do like, comment and share my video. So actually, digestion of food starts from our mouth. The buccal cavity of our mouth performs two major functions: mastication of food and facilitation of swallowing. So mastication of food means chewing of food. So first we chew our food and then we swallow. With the help of saliva, our teeth and tongue help in this mastication process. Okay. So the teeth and the tongue with the help of saliva masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. So saliva contains mucus which helps in this lubrication process. So basically, bolus is the mixture of masticated food particles and mucus. Okay. Now, after the buccal cavity, we have pharynx and esophagus. So, bolus is then conveyed into the pharynx and then into the esophagus by swallowing. Saliva contains amylase, lipase and other enzymes. Amylase breaks down carbohydrates and lipase breaks down lipids. Now 30% starch of our food is hydrolyzed by this amylase enzyme which is present in the saliva into a disaccharide maltose. So starch is broken down into maltose by the enzyme amylase, and maltose is the disaccharide, and this happens in the mouth. Lipase plays a major role in fat digestion in newborn infants. Okay. Next to the esophagus, we have stomach. The gastroesophageal sphincter is located at the junction between the esophagus and the stomach. It controls the passage of food into stomach. So, stomach contains gastric glands. Gastric glands contain chief cells and parietal cells. Chief cells secrete pepsinogen, renin and lipase, whereas parietal cells secrete hydrochloric acid. Okay. Now this pepsinogen that is secreted by the chief cells of gastric glands, it is converted into pepsin by this hydrochloric acid, just like that. Now this pepsin converts proteins into proteases and peptones. So food contains proteins, right, which are now broken down into proteases and peptones by the help of pepsin. And HCl provides acidic pH, that is pH 1.8, which is actually optimal for this pepsin. Renin of infants helps in the digestion of milk casein. Next is small intestine. So next to the stomach, we have small intestine. Bile, pancreatic juice and the intestinal juice are released into the small intestine. So bile is actually secreted by the hepatic cells of liver. And this bile contains pigments such as bilirubin, biliverdin, etc. Bile also contains bile salts, cholesterol, phospholipids, but it does not have any enzyme. Bile helps in emulsification of fats. Emulsification of fats, it means breaking down of the fats into very small micelles. Bile also helps lipase enzyme. So we have seen that small intestine also contains pancreatic juice. Pancreatic juice is secreted by pancreas. This pancreatic juice contains trypsinogen, chymotrypsinogen, amylase, lipase, procarboxypeptidase, nuclease, etc. Trypsinogen is activated by enterokinase. Enterokinase is actually secreted by intestine. So trypsin is activated by enterokinase into trypsin, just like this. This chymotrypsinogen is converted into chymotrypsin by the help of trypsin. Procarboxypeptidase is converted into carboxypeptidase by the help of trypsin. Now, this trypsin, chymotrypsin, carboxypeptidase, they are actually proteolytic enzymes. That means they break down proteins. So, proteins, peptones, proteoses. So, peptones, proteoses, these are made in the stomach, right? Now, these proteoses, peptones and rest of the proteins, they are converted into dipeptides by the help of this trypsin, chymotrypsin and carboxypeptidase. So, we have seen that 30% of starch was hydrolyzed in our mouth. Rest, that is 70% starch, is now hydrolyzed into disaccharides by the enzyme amylase. Fats are broken down by lipase. Triglycerides, which are called fats, are broken down into diglycerides, and diglycerides are broken down into monoglycerides by the help of the enzyme lipase. Foods not only contain carbohydrates, fats, lipids, but it also contains nucleic acids. So, nucleic acids of foods are broken down by nuclease. Nucleic acids are first broken down into nucleotides and then nucleosides, which are done by the help of nuclease. Next is intestinal juice. So, we have seen that a small intestine also contains intestinal juice. Okay. Intestinal juice is secreted by intestine. So, intestinal juice contains nucleosides, disaccharides, that is maltase, lactase, sucrase. It also contains lipase and dipeptidase. So, nucleosides are now broken down into sugar and base by the help of nucleosides. Maltose is now broken down into glucose by the help of maltase. Lactose is broken down by lactase into glucose and galactose. Sucrose is broken down into glucose plus fructose by the help of sucrase. Monoglycerides are broken down into fatty acids and glycerol by the help of lipase. Dipeptides are broken down into amino acids by the help of dipeptidase. So, this breakdown occurs in duodenum. Actually, small intestine is made up of three parts: the duodenum, jejunum and ileum. And this breakdown occurs in duodenum. Simple substances which are now produced after the digestion process, these are now absorbed in the jejunum and ileum, and the undigested and unabsorbed substances are passed on to the large intestine. So this is all about today's lecture. I hope you liked the lecture. Thank you for watching my video.